Good afternoon. My name is Terry Albarano, and I'm a pharmacist representing Omnicell's IV compounding automation portfolio, and I'm excited to welcome you to today's continuing education program. Omnicell and ARL came together to co-sponsor this continuing education program titled Rapid Sterility Testing Strategies for Compounded Sterile Preparations. Dr. Kevin Hansen and Brian Kelly declare no conflicts of interest. And again, I'd like to remind everyone this program does offer continuing education credits. At the close of the program, please leave your browsers open. You'll be redirected to an evaluation that you need to complete to receive your credits. Today's presenters are Brian Kelly, Director of Business Development with ARL Biopharma. Mr. Kelly is Director of Business Development with ARL Biopharma, and he graduated with a BS in Biology from Oklahoma State University. After receiving his degree, he worked 10 years for Genzyme. While at Genzyme, Brian's focus was on the development of novel therapeutics for rare diseases by performing analytical chemistry, cell culture, and preclinical pharmacokinetic and pharmacodynamic studies. 8 years ago, Brian began working in project management for ARL. For the past 4 years, he has been in his current role, Director of Business Development. And I'd also like to welcome Dr. Kevin Hansen, Assistant Director of Pharmacy at Moses H. Cone Memorial Hospital in Greensboro, North Carolina. Dr. Hansen provides leadership and operational oversight for pharmaceutical compounding and pharmacy perioperative services. He's also the Residency Program Director for the Health System Pharmacy Administration Residency Program. Dr. Hansen serves as adjunct faculty for the University of North Carolina Eshelman School of Pharmacy in Chapel Hill. And within Moses H. Cone Memorial Hospital, Dr. Hansen is involved in multiple committees and initiatives related to his practice interests of pharmaceutical compounding, handling of hazardous drugs, medication and compounding safety, drug shortage management, and pharmacy perioperative services. He has taken a lead role in developing multidisciplinary teams, such as the Pharmaceutical Compounding Advisory Council and the Hazardous Drug Committee. He also provides pharmaceutical compounding guidance and standardization across Cone Health, which is a 6-hospital health system. Before starting, I'd like to review today's session's learning objectives. These objectives are to define rapid sterility testing methods, compare rapid sterility testing method to traditional USP 71 sterility tests, outline factors to consider when implementing rapid sterility testing methods, apply rapid sterility testing strategies for compounded sterile preparations, and identify advantages of rapid sterility testing application in sterile compounding. We do have a list of abbreviations that are covered in the slides, and I'd also like to remind everyone that the slides are downloadable on the registration site. And with that, I will turn the presentation over to our speaker, Brian Kelly.
Thank you, Terry. Okay, so start with the question that we get a lot is, am I allowed to use rapid sterility? Rapid sterility test methods are considered alternative sterility test methods, and 797 allows for the use of them if the test method is validated. Okay, validated in the user's hands, not validated by the manufacturer of the instrument or the technology. And that validation must prove that the rapid sterility test method is non-inferior to 71. 71 is still considered the benchmark when it comes to sterility testing, so that validation for any rapid sterility test method must prove that that test method is at least as good detecting microbial contamination. So USP 1223 explains how to perform that validation. But there are a variety of additional resources available, including one from the Provincial Drug Association. So it's not hard to find information on how to perform the validation, but the validations are pretty involved. Another question we often get is, does the FDA accept rapid sterility test methods? The answer is yes. It's not a blanket approval for any particular rapid sterility technology, but they have accepted them on a case-by-case basis. And whether or not the FDA will accept the use of a rapid sterility test is dependent on if they agree with the validation. So there's a lot riding on that validation, and we'll talk a little bit more about it in a bit. So in short, a rapid sterility test method is any sterility test where test results can be obtained faster than the 14 to 18 days required by 71. Rapid sterility test methods can detect microbial growth faster than 71 because they use a different detection method. USP 71's test results are obtained by visual inspection of the growth media for cloudiness or actual growth. That's why the test takes so long. You have to allow time for visible growth. Okay. There are a variety of rapid sterility technologies available. These technologies have either A, allowed for shortening the 14 to 18-day incubation period, or eliminating it completely. For those technologies that have completely removed the incubation period, sterility test results can be obtained in as little as 2 to 3 hours. So that sounds perfect, right? Well, not so fast. Each of the rapid sterility technologies has strengths and weaknesses. To be fair, 71 has its strengths and weaknesses. So it's important to keep in mind there's no such thing as a perfect sterility test. And so during this presentation, we're going to look at the strengths and weaknesses of some of the rapid systems, and also talk a little bit about some of the strengths and weaknesses of 71. Here's a summary of some of the rapid sterility methods or instruments available. Again, notice if you look at the detection principles, they use a variety of techniques to detect microbial growth. Because they use different detection methods, each require different amounts of time to produce a result. Some of them I've listed here in the table as a range to the time to result. That's because the actual time to result is determined during that validation. Okay. Now we're going to take a closer look at a few of these rapid sterility test methods. I've divided the technologies into two categories, growth-based and non-growth-based. Growth-based technologies are similar to 71 in that they utilize growth media and an incubation period. It's just that the incubation period is shorter than 14 days. Non-growth-based technologies do not utilize growth media and incubations. These rapid sterility technologies have moved completely away from the traditional microbiological techniques. At least two of the growth-based systems that were listed in that table and that we're going to talk about are designed to detect ATP, so microbial ATP, as opposed to, again, waiting on the growth period and looking for a visual clue of growth, whether that's cloudiness of the media or actual growth. These technologies are designed to detect ATP from microbes. So you may recall from your biology classes that ATP is the energy-carrying molecule found in all living cells. So these tests use an enzyme that break down ATP to produce light. The light's then measured by a luminometer built into the instrument. The theory, the more light detected, the more ATP present, the more microorganisms are present. And one of the ATP detecting instruments is Milliflex by Millipore. As mentioned earlier, all these rapid sterility technologies, they have strengths and weaknesses. So a couple of strengths of this instrument are that the test results are assigned by the instrument. You get an actual number as a test result. So it doesn't require any interpretation by the analyst. So the result is objective, as opposed to some of them that we're going to talk about that are subjective. So another strength is that this test procedure is non-destructive. So in the unfortunate instance of a non-sterile test result, it's possible to identify the contaminating organism in the exact sample that was being tested. Identifying the contaminating organism in the case of a non-sterile result is an important part of a good out-of-specification investigation. And it's hard to fix a problem when you cannot identify the source of the problem. And to be able to identify the source of the problem, it's real helpful if you can identify the organism that is the problem. Okay. So that's the strength of this system. It's a non-destructive test. Weaknesses here, because it's a growth-based system, right? It takes time to grow up enough organisms to be able to detect that ATP that comes from the organism. So, because this is a growth-based system, it takes more time than some of the other technologies. Another weakness of this test is it can only test filterable products, so it can't test oils or suspensions. Another ATP detection method is the Celsus Advanced by Charles River. This system has all the strengths of the Milliflex system, plus it can test all sample types. The reason why it can test all sample types is because the initial setup is exactly the same as a 71. So if a sample can be tested by 71, the sample can most likely be tested by the Celsus Advanced. Weakness, again, is that it requires a growth phase, which adds time compared to the non-growth-based systems. The last of the growth-based systems we're going to look at here is the BacT-Alert 3D by bioMérieux. This one is a growth-based system, but works completely different than the ATP detecting systems we've just looked at. This one actually works by looking for a color change in the growth media, and the instrument's doing that on a continual basis. So one of the strengths here is that it can provide test results faster than either of the two growth-based systems, the ATP growth-based systems that we were talking about, because of that continual read. So the test results from this system can be obtained in 1 to 3 days, whereas those other two systems, the ATP detecting systems, take more like 5 or 6 days. Okay, but as I mentioned earlier, none of these systems are perfect. The weakness of this system is it can only detect certain types of microorganisms. So you may get a sterile result for a sample that's not sterile. So they all have strengths and weaknesses. Another one, weakness for this system is that direct inoculation is required. So direct inoculation, I'm not going to get into the details of that, but basically DI, or direct inoculation, increases the chances of microbial contamination to occur during the test procedures. So USP 71's preferred test setup is the closed membrane filtration, not direct inoculation, so not really following 71's preferred method there on sample setup. So a quick look at the non-growth-based systems. So rather than depending on time and detection of some biochemical or physiological properties, these techniques use cell labeling. So label the cell right away, use that labeling as a methodology to detect microbial contamination. Because of the way these work, they don't require growth periods, and they're the fastest test methods available. So the most common of the growth-based rapid sterility test methods is the ScanRDI, also by bioMérieux. So interesting, bioMérieux makes both a growth-based and a non-growth-based system. The biggest strength of this system is that it's fast. It's very fast. Test results can be obtained in about 3 hours. But weakness of this system is that the sample is destroyed during the test. So during a non-sterile out-of-spec investigation, you can't identify the contaminating organism in the exact sample that the organism was detected. True, you could go back and pull samples out of the rest of your batch of IV bags or syringes or vials or whatever, but microbial contamination is often not uniform. So just because it was detected in the vial that was subjected to the test doesn't mean it's going to be found in other vials. So that's a weakness and really limits the value of an out-of-spec investigation when you don't have the opportunity may not have the opportunity to identify the contaminating organism. Another weakness of this system is that the test results are subjective. By subjective, I mean that the results are obtained by analyst interpretation. So this is a cell labeling technology, but unfortunately the cell labeling will stick to things that are non-microbial. So an analyst looking at a computer monitor must decide if something that they're looking at that's glowing that has that label stuck to it is a microbiological cell or some other particle. So because it's open to this interpretation, then that can lead to incorrect reporting of either sterile or non-sterile results. So again, all these systems have strengths and weaknesses. So, but one thing all of these systems have in common is that method suitability is required. So method suitability, it's a formulation or product-specific validation. So during method suitability, that's where microbiologists determine how to overcome any antimicrobial properties of the drug. It's basically where they figure out all the details of how the test is going to be performed later on that specific drug product. So again, so USP 71 technology is validated, so only method suitability is required. But rapid sterility technologies must be validated and then must also have method suitability. So another layer there. So sterility testing on your batches, it's not only a good idea, but it's a requirement of 797. It's important that you understand that rapid sterility test does not in and of itself ensure sterility. Sterility test results are a lagging indicator that all the other things you're doing are working appropriately. So that your quality system is working appropriately, that it's robust and thorough, right? So things like staff training for garbing procedures and aseptic technique, along with continual monitoring of those activities. Speaking of monitoring, you also have your environment, right? It needs to be sufficient to detect any problems or lack of control in the compounding area. So the sterility test is an indicator to tell you if your quality system is working properly. Okay, a little more on USP 71. I mentioned earlier that the test results are obtained by visual inspection of the growth media. The visual inspection, again, is to look for cloudiness of the media or actual microbiological growth, but it's the visual read is not as straightforward as you might think. So looking at the cloudiness of the media in this picture, one might think that vials 1 and 3 are sterile and 2 and 4 are not sterile because you can't read the writing behind them. However, it turns out that vials 1 and 2 are sterile and vials 3 and 4 are not sterile. So there's certainly some subjectivity, you know, from one analyst, what they consider cloudy versus what they consider not cloudy. You know, is that growth? Is that not growth? So obtaining 71 test results requires expertise, but there is still subjectivity to the 71, which is still considered the benchmark. So as opposed to the 71 with its subjectivity of its test results, the systems that detect ATP, they provide an actual number, an objective test result. So a little bit more about how these systems work is they use the enzyme that makes a firefly glow. When that enzyme interacts with ATP from a microbe and another reagent, light is produced. So the more ATP present, the more light is produced, and an actual measurable result is obtained. So again, the test results from ATP detecting systems are objective, not subjective. So when deciding if you're going to utilize a rapid sterility test and determining if the technology is appropriate, you should consider how that test compares to 71, again, because 71 is the benchmark. So here's a table that compares 71 to the Celsis Advanced system. By reviewing the table, it can be concluded that the Celsis Advanced takes advantage of the strengths of 71, but improves on 71 by shortening the incubation period and providing objective test results. Okay, so with the faster test results, not only can batches be released faster, but any problems with the clean room or with compounding can be addressed sooner. Here are some other things to consider when implementing a rapid sterility test method, including knowledge and experience of the supplier. So the supplier could be the outsourced laboratory, or if you're testing in-house or considering it, does your staff have the ability to perform the test appropriately and interpret results for those that require interpretation, but also perform the validation, because again, that has to be done at the user level. But switching to a rapid sterility test method is a risk-based decision. But keep in mind that the riskiest decision of all is to not test for sterility or not wait till the sterility test is completed before releasing a batch. Okay, so after evaluating all the available systems, we decided that the Celsis Advance was the preferred system. We fully validated the system and proved it to be non-inferior to 71 as required by 797. Again, method suitability is still required, but the validation has been completed here. Please contact me if you have any interest in discussing the technology or the validation, but I'm now going to hand off to Kevin for his portion of the presentation. Thank you.
Thank you so much, Brian. So in my section, I'm going to really be talking about the benefits and also the implementation from a case study that we've done here at our institution. So I think it's important to kind of reflect back on why do we even perform the sterility test. And Brian said it really best is the fact of doing a sterility test does not ensure that our batch is sterile, right? But it does give us some assurance or confidence that the compounded batch is sterile. So what do I mean by that? So when you think about your sterile compounding operation, think about all the variables of things that go in that influence the sterility of that product. We're not just thinking about our hood, but we're thinking about, our people especially, their practices, their training, their aseptic technique, the cleaning, the maintenance of that, our HVAC system, all of these things, our product, our equipment, our environment goes into influencing that. So really, you ask yourself in the question is, do I have 100% state of control at all times during the compounding of every batch? And that's, really almost an impossibility to truly say that I have that state of control 100%. So performing a sterility test can indicate to you if one of these systems perhaps failed and you weren't able to detect that. You're going to be able to see that in gross contamination within a batch. And so again, it can give you a level of assurance or confidence, especially when you're doing potentially large-scale batching or sending out products to multiple institutions, or that can go out to many different patients. So definitely something to consider as we talk more about the benefits just in general of why we perform sterility tests on our sterile preparations. And again, I'll reiterate, patient safety is best served through the completion of a test that detects the microbial contamination prior to a product use. We know that USP 797 allows us or gives us the option to release a product before the results, but we know that's not best for patient care and something that we should all strive to do. So again, I think Brian touched on some really good points about some of the challenges with our traditional methods, and I want to just talk about this maybe a little bit more operationally of what this looks like in a practice setting. And I'll be referring a lot of this to my practice setting of thinking in a hospital pharmacy or a 503A compounding operation within a health system. So using our traditional methods, USP 71, it may be difficult to determine what that proper batch size is too far in advance on usage, right? Because we're having to wait at least 14 days before we can release those products from quarantine for patient use, which then relates into us having a tendency to produce these larger batch sizes, which then, of course, could result in expired or wasted preparations and drug going down the drain. A pressure to release preparations. And we'll talk a little bit more about this when our stock gets very low or is depleted. What do we do in those instances? Significant space is needed for quarantine preparation storage. So if you're performing USP 71, you're having to have at least 14 days worth of inventory removed on there for patient use. And so that could take up a considerable amount of space determining based on your operation size. And lastly is about a resultant shorter effective beyond use date. And so what I mean by that is how much beyond use date is remaining on the date and time that you take it out of quarantine and put those into circulation for patient use. And that may even prevent you from placing those in automated dispensing cabinets or in areas that would really be best to serve patient needs. In contrasting this with some of the benefits of rapid sterility testing, so really the opposite and a lot of those things that we just discussed, we know that we, of course, will have this reduced quarantine time while we're awaiting those results. We can decrease costly drug waste, right? If we have a longer period of time that our drugs can be used, we potentially will lead to less waste of those products. Decrease in internal shortages, again, turning over from time of when it's prepared to it can be used by a patient, so we can get those in much quicker and even potentially do another batch, knowing that we're going through or having high demand on a product. Reducing the requirement for our quarantine space footprint. So for the Celsis system, Brian discussed that having a 6-day period. So we can take the footprint for quarantine from 14 days down to 6 days. And so that may reduce that footprint of space needed. Then thinking about the worst case scenario of when, what happens if you did have a gross contamination that was detected in the system. Well, if you're able to detect that within that 6-day period, you can certainly start your investigation of that much sooner and even start a new production to get that out. So again, you're not creating shortages or gaps in products for your patients. And then lastly, going off this objective versus subjective measure is that we're going to have increased confidence in the results because we do have a very objective based test. So now we're going to shift over and talk a little bit about implementation and what were some of the considerations, right? So when we linked in with ARL and learned about all the benefits of rapid sterility testing and the tests that were performed, we said, hey, how do we want to proceed? We can either do one, continue just doing USP 71 for all of our products. We could do a hybrid where we're doing some rapid and some USP 71, or we could do all rapid. And so we have opted to do this hybrid model where we were very selective of what products we'd be using as rapid. And again, this is just an option. So the methodology that we developed when we first looked at all of the products that we were producing with this batching program using IV automation is we came up with these 6 different things that would determine, hey, maybe these are ones that makes more sense to have rapid sterility testing. So one would be preparations with relatively shorter beyond use dates, right, to help to maximize the usefulness of those to decrease some of that waste. Looking at our production schedule or the cadence, how frequently are you batching or producing those sterile products? Low on hand, ready to use inventory. Having a wide standard deviation of usage or demand on a product. Thinking about holidays and downtime delays, especially when you consider your hours of operation for your production. And then lastly, the expense of a drug ingredient. So for a drug that was really expensive, would we consider doing this to maximize that shelf life compared to something that was less expensive? So we'll go through each of these categories and talk about a little bit of examples of each of those to paint a little bit clearer picture about what this looks like. So starting with preparations with this relatively shorter beyond use dates. So let's consider a product that has a 21-day beyond use date, right, from the date and time of what it's compounded. So if we follow the USP 71 and assume that 14 days are going to be subtracted from that, you can see that removing that from quarantine with a successful result of sterile, we can see that we're only left 7 days. So when we're thinking about in the hospital setting, having only 7 days on a product may prevent me from placing it into my automated dispensing cabinets in my ICUs or in my operating room. And really, it's operationally, it can be very challenging to manage something with that short of a beyond use date. Contrasting that, if we were to use rapid sterility testing using the Celsis system through ARL, you can see that instead of that 14 days, we'd be looking at 6 days. And so for that same product where we had 7 days before, we'd be looking at 15 days. And so as we consider that, 14 days could certainly change operationally what we're going to be doing with those preparations. And again, just visually talking about what I mean by effective beyond use date. So if we were to use, for example, just a preparation that had a 60-day beyond use date, and we're following a USP 71 sterility test, we have to think about all the time that goes from the date and time it was prepared to that moment in time that we release it. And we generally will use or estimate about 15 days, since we do lose a day of shipping out the samples to ARL for testing. And then, of course, we lose the 14 days to the incubation. So total of 15 days that these products are in quarantine. And so when it comes out, we'll actually calculate, what is that percent remaining of that labeled beyond use date. So for this instance, it would actually be 70% of the labeled beyond use date, which would be 45 days. Next is on the production schedule, or I'll say the cadence. So, if you were looking at a preparation that had a 60-day beyond use date, you might, think, oh, if it's 60-day beyond use date, that means that we'll just make one batch every 2 months. Well, operationally, that's always not the case. There are several variables that go into what is your cadence and what is your batch sizes that you create. So you got to think about your product usage. You got to think about your batch size limitations. So whether that be staffing or automation efficiencies. Potentially, you couldn't even produce a certain batch size just given the current efficiencies with certain things. Thinking about the preparation beyond use date, of course, hours of operations. Are your batching program, is it running on weekends or holidays or overnight? Thinking about how much storage space you have. I may be able to make, 1,000 of a product, but do I even have the storage space to hold all of that? And then lastly, the availability of the ingredients and materials, especially through drug shortages and allocations, that can be challenging. So there may be times operationally where you have a product that has a 60-day beyond use date, but the cadence of production is every 2 weeks. So you can imagine in this instance, if you were waiting 15 days to get a sterility test result, it kind of puts you in this pickle where you're, releasing product, but you're also waiting on product to go out to adjust the batch size, and kind of puts you in a rear with production and can cause some shortages. Then the lovely situation of what do you do when you have low on-hand ready-to-use inventory? Let's say you had increased demand since your last batch, and your frontline staff come to you and say, hey, our stock is getting low on this, preparation. When's the next release date, right? And you think, ooh, we literally just batched that. We got about 14 days left, right? So then my quality assurance coordinator is scratching his head saying, what do we do, right? Our policies and everything says we don't release from the results. So we get into these tricky situations. And so again, these may be instances where if this is happening with a certain product line, you may want to consider switching it to rapid sterility testing to help ease some of those concerns. And that goes hand in hand with the next point on the standard deviation of usage. So at least in acute care within hospitals, for some products, we do see some seasonality of usage, right? So when we think about our batch sizes, we're not just using averages. We may have a flexible par level, or we may be looking at the last month and trying to estimate what that usage may be. So for this product, looking over a year, we use anywhere from 500-ish on the low end all the way up to almost 900. So what is your batch size that you create? So if you're able to have products released sooner and you're able to create maybe smaller batches in between, you can reduce the waste and expired product and decrease some of those stockouts. And then thinking about holidays and downtimes. So, especially when we have these extended holiday weekends where a holiday may fall, a day before the weekend, you got to think, can my operation support having 4 or 5 days removed from my beyond use date or before I can even get that product into circulation, and can make it very challenging when you're going through and having products that are in high demand, but you're having batch size limitations there. So you have to consider your operations as well. And then lastly is the expense of the drug ingredient. So when we talk about waste, for instance, one of the products that we prepare is Sugamidex syringes, and we know that the cost of one of these vials, the average wholesale price is a couple $100. And so we really want to do everything we can to minimize those products from being wasted, and that's through maximizing of those beyond use dates and adjusting our par sizes and getting them to the correct locations and the correct amounts. So now we're going to talk about a case study, and we're going to follow a single product line. So we'll talk about our Sugamidex syringes that we created, so you can really start to see, operationally, what does this look like in practice, right? We heard a lot from Brian about what the technology does and our incubation periods and all of those specific details, which are great, but what does it actually look like in practice? So I can share, we've been doing rapid sterility testing since April, and so we have some data that we can certainly share. And so this case study of looking at these Sugamidex syringes, we're starting with these 5 ml bulk vials of Sugamidex, and we're running that through our IV robot, our IV automation, and we're packaging those into these 2 ml ready-to-administer syringes. So you can see here what that looks like in our labels, and those get that 60-day beyond use date on those preparations. And I think it's important, especially while we're talking about sterility testing is, well, Kevin, you're taking a sterile product and just, drawing it into a syringe, why use an IV robot? Wouldn't you want that for compounding instead of repackaging? But I think it's important to say, again, that the sterility test doesn't increase necessarily the quality of the batch. You have to look at the method of preparation. And so when I look at an IV robot and you look at sterility assurance, USP Chapter 1211, right, and it really underlines this point that operating personnel are the most significant contributors of microbiological risk, right? Further, then that underscores the importance of why we should think of separating personnel from that aseptic environment and limiting their interaction with these sterilized components and products. And so, when I read this and read this chapter here, I instantly connected that with IV robotics because it is a restricted access barrier system. It's an ISO 5 cell that humans don't have interaction with, right? We're loading the device, the arm is taking it into that, and during those critical transfers, a human is not present. And so that's why in our program, we only batch through these IV robotics devices. So thinking about our highly oversimplified workflow, of course, step one is we will compound or aseptically draw these products using our IV robotics. Those preparations do all get visually verified by our quality assurance coordinator, and they look at some documentation and they look at everything to ensure that it is accurate as is. And throughout the life cycle of that compounding process is we're taking samples. So we're being forward thinking and knowing, hey, we're making a batch size or targeting a batch size of 250 syringes. So we know that we need at least 10 samples pulled from this batch to send out to ARL for testing. And so there's a couple different ways you could think about it is, where do you take those samples from? Do you take those all, the first 10 that it produces? Do you take the last 10 samples that were produced? Or do you take some from the beginning, middle, and the end? So there's a couple different ways you could think about it, but really ideally is you would want to probably is get a represented sample kind of scattered throughout. So you have a really good representative sample of that entire compounding process of when this batch was produced. So then we're taking those 10 samples, we're shipping those to ARL, and the remainder of that batch is going into a locked box with documentation, and that goes away from our dispensing environment into our quarantine locked bin process. So then ARL is performing the rapid sterility testing using the Celsis, and then after 6 days from when the testing starts, we get those final results. Our quality assurance coordinator will review those, and then we make a decision, do we release them or do we not release them when we investigate? If it's sterile, then we will release those products for patient use. So as Brian mentioned, how many samples do you need to send? And for the study, the non-inferiority study that was done by ARL, they tested it using the same number of samples as USP 71. So this actually made it very easy for our staff because we have been doing sterility testing with this batching program for over a year. So we were very familiar with this table in USP 71 about how many samples need to be tested. And so we didn't have to think, oh, is this rapid, is this not? How many samples do we need? We just always keep the number of samples the same. And so this is here for reference. Next, I think it's really important for folks that are doing a batching program is to have a really clear definition of what you consider a batch size. And so we discussed this and had our own opinion here at our hospital, and we actually engaged with our State Board of Pharmacy, which I encourage others to do as well. Is to say, when you come and inspect, what are your expectations? What do you would consider a batch size? And we had a great collegial conversation with our state board, and we came down with the following: that each batch would be individually considered based off of the compounding equipment, the ingredients, and then the day of preparation. So what does that mean? So we have 2 IV robots, and we're in the process of adding our 3rd IV robot at this case. But each robot produces its own individual batch. So we would not have a batch that would separate over 2 robots. Again, the thinking is if there's a failure, right, and Brian was talking about the investigation, we would want to be able to go back and say, hey, there's something wrong, right, with this robot, and we would evaluate that practice. The other way to think about it, if there was a sterility testing failure and you did separate it out, you did not separate it out between, you risk potentially losing double the amount of products as opposed to half. So it's kind of a trade-off, right? You do more sterility testing, but it also increases your ability to have a better investigation process. So something that the institution should discuss. We also will do it to make sure at the ingredient level, all of our ingredients have the same lot number. So if anyone's been in an operational pharmacy and knows what it's like to have a recall on a drug and try to relate those back to those individual patients, it's extremely difficult, if not impossible. So we actually have within our dispensing environment 2D parsable barcodes, so we actually can link an individual NDC or lot number of a product back to a specific patient that it was administered to. And so again, this is our way of really connecting back, and if something does go wrong, we can have a very robust investigation process. And then at the day level, each day we do cleaning, of course, per USP 797. And so we did not want to have batches stretch into multiple days because we know that there could be different environments or even different cleaning practices. And so these are the three variables that goes in to determine what our batch sizes look like. So as we factor in all of those variables and we look at weekends and holidays and shipping and all of this, we started with our USP 71 sterility testing of having a period between 15 and 19 days that we were actually releasing products from quarantine. And for the select few that we have been doing rapid sterility testing, we've gotten that down between 8 and 12. And this actually will be slightly less upcoming. This is obviously during the period of our COVID pandemic. And so we know that some of that has been impacted there as well. So just to drive this home point, let's go ahead and we grabbed one of our logs from 2019 so we could follow this product and compare it. So here's using our traditional USP 71 sterility testing, and we intentionally chose one that was on a Thursday production so we could really show you a worst case scenario of how these weekends and things can impact what that effective beyond use date is. So Thursday, September 12th, we produced these 272 units of these Gamodex syringes. We've extracted 10 of those syringes that are going to go off for sterility testing, so 262 of those syringes are going into quarantine. And our beyond use date at this point, obviously, is 60 days, and we have that date there. It's not uncommon for our production that production will last most of the day, so then they actually get verified by our quality assurance pharmacists on the next day, so now we're into Friday, right? And then on that day, after everything is signed off, they'll go ahead and take those samples, package them up, and those will get shipped off. So then we can see that we lose a day for shipping through the weekend, and now we're at Monday of when ARL will receive those samples, and they're going to go ahead and test those on the same day as well. And then fast forwarding another 3 days, we do get our no growth at 3 days observed. So that's one of the benefits, I guess, of 71 is you do get preliminary results. However, we don't do anything differently with those if there is no growth identified. And then we get our final results on Monday, so now we're up to day 18. So we can see that we've gone from 60-day beyond use date, and now we're at 42 days of that effective when we take it out of quarantine, which is 70% of that original beyond use date. And quickly here, comparing this to rapid testing, and we picked one that was a real-life scenario of doing it on a Thursday to see what the comparison would be. And we go all the way down here, and we can see that after 12 days is when those were released, which is 49 days of their remaining beyond use date, which is 82%. So we can see that we've taken what I call the 30% effective beyond use date loss to the quarantine process and reduced that down to 18%, or essentially gaining 7 days additional back to our beyond use date of those products. And again, Gamodex is not an inexpensive drug, and so that's going to relate to decreased waste, which is excellent for us. All right, so looking at some of our data, so we've been in operation for a little over a year, so we've done over 750 batches, and we do correlate each batch one-to-one with the sterility test, so we performed over 750 sterility tests with ARL, and you can see back in April is when we started doing our rapid sterility testing and continue to do this. July looks shorter just because that was a partial month, but we expect that to be increasing or on par with those previous months. So knock on wood, to date, we have not had a single sterility testing failure. And again, this is something we're proud of. and have good practices in place of knowing what happens when something does fail. But again, if you focus on your method of preparation, of which we use those IV robotics and those restricted access barrier systems, you should expect this. And so something that is really important to consider. Looking at all the products that we produce and the number of batches that we produce, you can see the lines with red are the specific drug protocols that we run through the rapid sterility testing. So a lot of OR syringes such as succinylcholine, Sugammadex, labetalol, and rocuronium specifically are the ones that we're running through the rapid sterility testing. So for our institution, we do in reading these chapters within USP and supporting literature about rapid sterility testing, it said really the institution should have a justification of why are we not using a USP 71 test. And so for all the aforementioned reasons, we sat down with the 4 products that we are doing rapid sterility testing on and said, well, why did we choose that? So for Sugammadex, as we saw through this case study, we had a relatively shorter beyond use date than some of our other products, and then also we had a very high drug cost. When we look at succinylcholine, that also had a relatively shorter beyond use date, but we had a very high standard deviation of usage, which correlated with a higher expired or wasted preparations. Looking at rocuronium, that was the same with the standard deviation of usage, and labetalol as well. So those were the specific reasons why each of those preparations were chosen for rapid sterility testing. So in summary, some of the take-home points, I know we covered a lot in a short period of time, but one, rapid sterility testing methods are useful for certain compounded sterile preparations that you've seen and learned here today. There are a variety of rapid sterility testing methods available, and each has their specific strengths and drawbacks. Several factors should be considered prior to implementing rapid sterility testing methods. And then rapid sterility testing can provide operational and financial benefits within a hospital and other settings. Thank you for your time.
Okay, as a reminder to everyone, please keep your browser open at the conclusion of the webinar so you can get redirected to the evaluation site for your continuing education credit. We've got a few minutes left here, so we will go through some of the questions that we received. We will provide a written response and send that out to all attendees for all the questions that were asked. One of the questions here was, how long did it take to validate the Celsis instrument? So for us, it took about 11 months. That's 11 months using multiple very experienced microbiologists. Of course, there's a couple of bumps along the way that should be expected anytime you launch any kind of new technology. But proving the non-inferiority to a 71 with the robust validation, including stressed organisms and everything that's involved in the validation, it was an 11-month process for us. Question for Kevin. Have you seen the benefits of using rapid sterility testing during the COVID-19 pandemic?
Yeah, that's an excellent question and definitely a benefit that I did not outline in this presentation. But one of the limitations, I guess, going back with USP 71 and with our operations, we run our compounding operation Monday through Thursday for the batching program, is because we know we're going to lose those 14 or 15 days for 71, we're shipping products every single day to be sterility tested. So that's a lot of shipping boxes, and that means that I have to have a pharmacist here every single day dedicated to checking those products. When we actually looked through this COVID-19 pandemic, we had some, especially early on, some reduced staffing and just shifting of resources. It actually allowed us to do batch checking. So potentially where we were not shipping products out every single day, but maybe we were only shipping them out once or twice a week. And we were still seeing, or we were not seeing, a reduction in our beyond use dates that we were seeing prior to USP 71. And so that's definitely a benefit of just thinking operationally, how are your products getting checked and how often are you sending them out? So that actually was very key through this COVID-19 pandemic and definitely something that we'll have in our back pocket now that we have the method suitability completed for those preparations. So great question.
Good. We've got a couple of different, I think I worded this differently, but the same question was, what's the difference, cost difference between a rapid sterility test and a traditional USP 71 test? That's going to vary some, but in general for us, it's about an additional $100. Let's see, we've got a question of, can you update the group where USP is with regards to the use of rapid sterility tests for release of products? Well, as I mentioned in my portion of the presentation, that USP does allow for the use of alternative sterility test methods, and rapid test methods are considered an alternative test method. And then they have USP 1223, which tells you how to validate that test method. In the 797 that they released last year, but then got pulled back, they did change the language a little bit related to the non-inferiority. I can't remember exactly how they changed it, but it wasn't a significant change. And so, yeah, as far as USP is concerned, I think they buy into the use of rapid sterility test methods for product release. I think then that also kind of hits a question: do you anticipate any of the 797 revisions regarding extending BUDs for CSPs affecting your current practice, the products you produce, and the BUDs that you assign? Yeah, a lot of that still has yet to be worked out with the BUDs. They didn't allow for any BUD extension in that revision, but that got pulled back a little bit, and I think that's part of what USP's currently looking into. So a couple different questions about the method suitability, cost, and time. So method suitability, there's a little bit more involved in the method suitability for a rapid system than what there is for a traditional 71 system. I guess it depends on what the rapid system is, but for us, there's several tiers available, and some of it actually depends on whether or not Omnicell has already performed a method suitability. So the cost from that can vary widely from $50 to about $1,500.
Yeah, I think to expand on that too, I think, within the question, it is an investment, right? And I think, in my portion of the presentation of really specifying to say, what specific preparations, do we want to do this on? And was a reason why we didn't say, hey, we're going to do rapid sterility testing on everything. It may or may not make sense for certain preparations. And so that's why we provided the methodology today to say, maybe here's where some areas where you may want to consider where it makes sense for that investment. And certainly with regards to this presentation, I see some other questions here about some other tests. So, for the scope of this presentation of really focusing specifically on sterility, but folks who are doing extended stability, certainly there's many other tests that need to be considered, whether that's container closure integrity, particulate matter, and other tests that are recommended. So certainly looking at USP monographs can help to outline some of those specific tests that are required for a preparation, but again, for the scope of this presentation was solely focused on testing that of sterility.
Kevin, do you see any others in here that you want to address during this time?
I see one other one where I think I understand the question they're asking. It's actually a great question, right? Of when you send something out for shipping, are you putting it on ice packs? Are you freezing it? That's something that we looked at and why we actually send out everything next day. and why we now with rapid sterility testing is determining when we send something out, so we don't have it sitting on a loading dock or through a weekend. So we've been a little more intentional like that with of when we're shipping products and knowing of when it's going to be received to decrease any opportunity of the environment having any impact on that. Certainly we're in North Carolina here, and so sometimes it can get very hot and humid out. So that's definitely something that that needs to be considered. So next day shipping and just considering of when you're shipping are two variables that that we've used. We have not gone into shipping the product frozen yet, but it's certainly definitely worth exploring.
I guess earlier on a question I took that I only answered part of it. One was how much does method suitability cost, and the other part is how long does it take. And then we had another question come in about what's the sample requirements. So generally the method suitability is going to take 2 weeks if the entire suitability is required here. And so, yeah, it is something that will need to be planned in advance and squared away. Also, the sample requirements, that'll vary a little bit, but in general for us, it's 4 times what your normal submission would be. So in Kevin's example of the Sugamidex, where he was producing a batch size of approximately 300 or so, and then he was quarantining 10 for testing, for us we would require, in that case, we would require 40 syringes for the method suitability if full suitability is required. Okay. Well, as I mentioned earlier, we will provide a written documentation of the submitted questions and our responses. We'll try to get that out within the next week or two. And thank you very much for your time, and be sure to keep your browser open so you'll be redirected to the site to get your CE credit and perform the evaluation. Thank you very much.